Hi, everyone. We're going to continue our discussion about eukaryotic microorganisms, and we're going to go talk about the fungi. And most of you are familiar with something about fungi. If maybe one of the first things you think about are mushrooms, and we think about the macroscopic fungi, mushrooms and puffballs. If you've been out and about, you might have seen a puffball. And so these are macroscopic. We're not going to talk about these in our microbiology discussion, but much of what we're going to say about fungi, about the microscopic fungi, is true for the macroscopic fungi. The fungi that we're going to talk the most about are the yeast and the molds. And in here, your the book describes it as the cells of the microscopic fungi exist in two forms, yeasts and hyphae. Yeast are round to oval shaped cells, and you can look at them under the microscope. Because they're eukaryotes, they tend to be larger than bacterial cell. And and the hyphae are kind of these microscopic thread-like or hair-like filamentous structures. And when you look at a mold, you can see those hyphae that are making up the mold. When you get enough of the hyphae together, you have something we call the mycelium. And that's a mass of hyphae. And we're going to talk about that in this lecture. But that's what you notice when you see a mold. But these molds are all around in the microscopic form before you can see those big colonies. Another form that's common in nature and important in disease is what we call a dimorphic fungus. And a dimorphic fungus is one that can be found in a yeast form or a mold form. And some of the fungi that infect the body might be things like they're growing on soil or feces or just out in nature somehow, and you inhale it because they kind of break off and fly through the air, float through the air. And when they get in the body temperature triggers them to form the yeast form, and they're more of like a yeast inside the body. So these we would call dimorphic. They have two forms. A little bit about fungi in general. When you look at mushrooms and things, they kind of look like plants, but they're totally different from plants because they're not photosynthetic. They don't have chlorophyll. They don't have a vascular system. They don't have like xylem and phloem that moves nutrients through the organism, and they more absorb their nutrients from the environment. And there's a couple other kind of unique things about fungi, and one is in their cell walls they have something known as chitin, chitin, chitin. And there was a picture of some chitin in a wall on the previous section of the book. But chitin is this molecule, is carbohydrates. It's in the wall of fungi, and it's the same stuff that's in the um, the shell of things like crabs and lobsters that makes that hard, and also insects. The exoskeletons made of chitin, and that shows you that fungi are actually a little more closely related to animals than they are to plants. The other thing is, just like every other eukaryotic and prokaryotic cells, fungi have plasma membranes, and there's an NCLEX question down here that refers to that. The plasma membrane, remember, this is the same thing as a cell membrane. And remember, the cell membranes are made of phospholipids and proteins, but they also have cholesterol in the membranes. Well, some fungi actually have a unique a unique sterol in the membrane instead of cholesterol. It's called ergosterol. And the reason why I mention things like chitin and ergosterol is because when we're looking for drugs to treat fungal infections, we have to find things that are unique in the fungi. So there's drugs that target their cell membranes, and there's drugs that target the synthesis of chitin that can be used to treat infections in humans. Okay. I wanted to talk just a little bit about the difference between molds and yeast. And so if we look at some of these pictures down here, we'll come back to that one. Here's a mold. This is kind of a picture of a mold and that fuzziness that you see in molds. And remember, we said they're made of these hair-like structures called hyphae. So this is highly magnified. These are actually microscopic. And so molds are made of these hair-like structures called hyphae. And they give you more details here than what we're going to talk about. Yeast, on the other hand, function, and these hyphae are always kind of in these big masses, okay? But yeast are found as single cells quite frequently. And so again, remember they're eukaryotic cells, and because they're eukaryotic, they have a nucleus. They're a little bit bigger than bacterial cells. But when you're looking at them under the microscope, one of the things you might notice is that they have these little baby cells coming off of them if they're actively growing, and we call them buds. If you were a bacterial cell, you'd have a bacterial cell. If it was going to divide into two, it would pinch off and it would divide into two equal size cells. Yeast, on the other hand, get these little babies on the sides that we call buds, and so they kind of divide unequally. So if you were to look at yeast under the microscope, they'd be really easy to tell from bacteria. You'd see that they were bigger, they have a nucleus, and if they were actively growing, you'd see these little buds forming. Let's see. I want to talk a little bit. I'm going to skip around here. I'm going to talk a little bit. Actually, let me show you this. Here's that NCLEX question. A patient with aspergillus, which is a fungal infection, takes an antibiotic, and it's talking about what part of the cell is it going to target. And so you could have figured that out from the discussion we just had. I wanted to talk about some unique features of fungal nutrition. So I'm going to go down here. So we're talking about how are fungi different from other organisms? Well, they're not autotrophs. They're not able to use photosynthesis because they don't have chlorophyll. They're like us. They're heterotrophs, and they're like most bacteria in that they have to acquire their nutrients from organic matter. And where fungi get their organic matter is frequently from dead and dying materials. So organic matter in the anywhere that there's decomposing organic matter, you're going to find fungi being major decomposers in that environment. So the term saprobe is a term for an organism that decomposes dead and dying material, dead and dying organic matter. Some fungi are also parasites, meaning that they can live inside of plants or animals, and they're living on or in that plant or animal, and they can make enzymes to decompose various things related to that animal. The other thing about fungi is here they mention that they have enzymes for digesting an incredible array of substances. A lot of things in nature, even leaves, are difficult to decompose. You have to have the right enzymes. And fungi make unique enzymes that can decompose things like leaves and feather and hair. And so they are really important decomposers. The other thing it says they're often found in nutritionally poor or adverse environments. Think about like the inside of your shower. If it gets kind of dirty, what you're going to notice is, or in between cleaning, you might notice mold growing there. You don't see other things growing there. You don't see bacteria growing, bacterial colonies. You see mold colonies. And it's because mold can survive or mold, fungi in general, can survive when there's very little nutrition and even just the slightest bit of water. So fungi are pretty good at growing in lots of different places. Down here, I talk a little bit about the concept of how hyphae form mycelium. And here it talks a little bit about that fungi can grow in colonies like bacteria do in a natural environment or on an agar plate. Yeast colonies look a lot like bacterial colonies, but mold colonies look fuzzy. And remember we said that the molds are made of those hyphae, those hair-like structures, and when you get masses of them, we call them mycelium, so masses of hyphae. When you're looking at a mold colony, you're looking at the mycelium, or masses of hyphae. Let's see. I wanted to talk just a little bit about how molds can propagate. And so when you have a colony that is a mold colony, or even if you're looking at your, bread that's got mold on it, there's going to be hyphae growing on the surface. And one of the things that the hyphae can do is the hyphae can just break off and float away. So if you have and they can make new molds. It'd be like if you could break off a finger and make a new person. Well, we can't do that, but molds can do that. They can break off a piece and start a whole new colony. And so when you have mold in you've got bread with mold in a bread in a bag, moldy bread in a bag. I have person in my family who loves recycling. So they always take the moldy bread out of the bag and put it in the compost and then take the bag and put that in the recycling. And I'm always saying, leave the moldy bread in the bag, because what you're doing is as you disrupt that mold, you're breaking off the hyphae, they're going into the air and they're available to cause mold to grow on other things. The other way that molds can reproduce is by spores. And they talk about spores here, and there's some pictures of some microscopic, here's some hyphae with some spores forming. These little packets have all these little spores in there. This one up here also had some pictures of some hyphae, and then spores are growing. And these are microscopic. When the molds have different colors, it's because they've started producing spores. The spores are also reproductive structures, and they can float away in the air and spread mold. So anywhere that you have dust, you're going to have mold. Just because mold spores and mold hyphae are really light and really small, and they can float around with the dust. I wanted to talk about some of the pros and cons of fungi. And so here it says that fungi pose problems to the agricultural industry. They can be very damaging to plants. There's a lot of fungal infections in plants. And think about, in your garden, you might have a blight, tomato blight, and that's caused by a fungus. And these fungi just live in the soil and they can survive from year to year. We used to have chestnut trees all over the East Coast. In the early 1900s, it was a dominant tree in many of the forests like what we live in Corning. And they're no longer here because there's a fungus that attacks the chestnut trees that causes chestnut blight. So there are really serious problems with fungal infections in crops. If we have in plants, if we have a fungus that causes a fungal infection, we call that infection a mycosis, or fungal infections in general are called mycoses, plural. So there's a lot of mycoses associated with plants. Right here, we also have fungi that are really important in decomposition, we said, in nature, but they decompose a lot of the food that we eat. It's estimated that 40% of the yearly fruit crop is damaged by fungi. But there's lots of benefits to fungi, and some of that decomposition is absolutely essential. If you're looking in the forest, you have, trees in your yard. We have trees all over the place. If we didn't have fungi decomposing those leaves, we would be buried in miles and miles of leaves. So fungi are essential for that normal decomposition on the planet. We also have a lot of these mycelia, a lot of these hyphae all through the soil, miles and miles of hyphae. One of the pieces of data I heard was under your footprint in a forest, under that space, there's about 3 miles of hyphae of fungi. And these are special kind of fungi called mycorrhizae that help plants grow. So we're dependent upon those for the health of all the plants that grow. Most of you are familiar with the idea that fungi are important in food production. They're also, such as beer, alcohol. They're for making bread. They're involved in making cheese and lots of other foods. We also use fungi to make antibiotics, different kinds of organic acids and vitamins that we use. So because they make so many unique enzymes, they make unique products. Um, the just to talk a little bit more about mycoses, which are the fungal infections. Your book goes into different ways that humans can acquire mycoses. I wanted to talk about this concept of opportunistic infection. Turns out that, there's lots of different kinds of fungi. Some statistic. There's lots of different kinds of fungi, and only a fraction of those, maybe less than 10%, actually cause diseases. Here we go. Nearly 300 species of fungi can cause, but there's nearly 300 species that can cause human diseases. But that's a small percentage of the fungi that we know about. The opportunistic infections are really common for fungi. What an opportunistic infection is, and it can be caused by anything, but fungi are usually involved in that. In the fungal infections often are opportunistic infections. What it means is these are low virulence species, meaning the pathogen isn't a real powerful pathogen. You have to generally already have something wrong. You already have to have a weakened immune system, or maybe be, sick in some way, or elderly, or compromised in some way for these organisms to make you sick. So we don't get quite as many fungal infections as we get bacterial infections because so many of these fungal infections are what we call these opportunistic infections. They're low virulence. You have to already be compromised in some way. I wanted to, they gave you an example of an infection that I thought was kind of handy to know about because it says uh, the disease I want to talk about is vaginal candidiasis, which is a vaginal yeast infection caused by the overgrowth of Candida albicans. And I think somewhere in here it talks about that 3 out of 4 women have had vaginal yeast infections, and some women get them chronically. And, Candida albicans is a yeast that most of us carry as part of our normal microbes in our body. And but no, it doesn't usually overgrow. And women know that if it does overgrow, it can be very itchy and very uncomfortable. And and you need to get rid of it. But what happens, it generally happens when your microbiota in your woman's vagina gets out of balance. And that can happen when they're ill from things like diabetes and other diseases. It can happen from taking antibiotics where you kill bacteria in the vagina and it allows the yeast to overgrow. So Candida albicans would be acting like an opportunistic pathogen, that it's not just because you're carrying Candida albicans doesn't mean you're going to get sick from it. Typically something else has to happen. A lot of women will find that when they eat a lot of sugar, it causes yeast infections because the sugar ends up in the secretions in the vagina, and that favors the growth of yeast. So, being aware of that and that Candida albicans is a yeast infection and can help you understand a little bit about how an opportunistic fungal infection can occur. The last thing I wanted to talk about was just a little bit about fungal. Oh, here they talk a little bit about another opportunistic infection. I'm not going to go over that one, but you can look at that and review that. Oh, actually, the one NCLEX. I'd mentioned that NCLEX question. The last thing I wanted to say about fungi and mycoses in particular is related to this medical moment here. And it asks you why is it difficult to treat fungal infection without doing harm to the host? So remember, we were trying to look at what about fungi are different from the host cells, and we said, oh, they have chitin in their walls, so you might be able to find a drug that targets their wall. They have ergosterol instead of cholesterol in their membranes, so you might find a drug that can target those. But remember, fungi are eukaryotic organisms. So when you are trying to find drugs that damage fungi, fungi are very similar to our own cells. So it can be very difficult to treat fungal infections, even things like nail infections, nail fungus. Um, for some of those nail fungus treatments, you have to get regular blood tests to see if they're damaging your liver. These are toxic chemicals. And the reason is because fungi are eukaryotes as well as us. So many of the drugs that are going to affect fungi and stop the growth of fungi might have high toxicity to other eukaryotic organisms. That's where I'm going to stop on fungi.